Yeah. Hey everyone, welcome to a new academic year of the data learning seminars. The first one this year is Marta Varela. She's a research fellow working in medical imaging at the Imperial College London. She did her PhD here as well. And yeah, thank you so much for doing this this first talk of the year, Marta. So go ahead.
Oh, my pleasure. Thanks very much for the invitation. And I'm looking forward to sharing my research with you all. I just wanted to say that I'm really happy to be interrupted if you have any questions as I go through. I probably can't see raised hands or anything, but feel free to unmute and ask questions. So I'm going to be talking to you about physics-informed neural networks and their applications in cardiology. So very briefly, I'll talk a little bit about the traditional way of studying the physics of the human body, which is using biophysical modeling, and then about PINNs, the physics-informed neural networks, and how they can be used to solve problems in cardiology, namely on cardiac electrophysiology, and they can be used as a solver for differential equations, for parameter estimation, and also using experimental data, and I'll tell you a bit more about it. At the end, if I have time, I'll show you some synthetic data that I've been creating using a cardiac MRI simulator with the aim of using it as training data for the PINNs, although I won't show you that data yet. So very, very briefly, if you were randomly asked to be a scientist and figure out how the human body works, I would imagine you'd have two approaches. If you had a very small, well-curated amount of data and a deep knowledge of the physics or the biology, in this case, of the system, you could design a model, typically using differential equations, and solve them and use that to explain how the human body works, normally in health or in disease. Alternatively, you could have huge amounts of data and not much knowledge about the physics and the biology of the system, but you could still feed all that data to a neural network or equivalent to make your desired inferences. Both these approaches, of course, have their pros and cons. I'll stress a few. The biophysical modeling gives you quite a deep understanding of what is going on in the body and what the disease processes are, compared to the neural networks, but the neural networks can solve a much wider range of problems. You're not constrained to the ones for which you know the equations. The biophysical models are very, very slow to run. You need to solve differential equations, as I was saying, and as you know, you need small time and space steps for them to converge to your solution. Likewise, neural networks are also slow to learn because they need to learn from large amounts of data. Biophysical models are hard to validate. Very often you create them making some assumptions, and it's very hard to check whether those assumptions are valid. Likewise, neural networks can perform badly when applied to unseen data. So focusing on the biophysical modeling for a while, and then I'll move on to the other approaches. There's a long tradition of understanding physiology and pathophysiology using physics. I chose this as the example. These are Hodgkin and Huxley, who won the Nobel Prize for Medicine and Physiology, seen here with what seems to be a huge lobster, but apparently is a squid. And they were doing experiments on the giant neuron of the squid, which you can see macroscopically, and acquired a lot of data about the potential difference across the neuron wall, which you can see here. So if you give a small stimulus to this electrical stimulus to this neuron, the potential difference across the neuron wall can undergo this nonlinear deflection, so a depolarization going to a higher potential, then a repolarization back, and an overshoot here, and then returns to the resting state. And during this period, which is the refractory period, you cannot excite it again. So this was all the data that Hodgkin and Huxley found out. They acquired more data such that they were able to create a model, a biophysical model of what was happening that led to this action potential, to this nonlinear deflection of the membrane potential. So if you like, electrical circuits, this is the electrical circuit version of their model. The phospholipid membrane of the cell is modeled as a capacitor, and then the protein spanning the membrane that selectively let ions in and out, they'll work as variable resistors and coupled with generators. You can also express this as a differential equation. The change with time of the potential membrane is given by this combined sum of what's called this ionic model. And you can extend this and use a similar model to study other excitable cells, namely cardiac cells in the heart. So we all think of the heart as a pump to send blood around the body. And if we forget that it is often sometimes that it is an electric signal that makes the heart contract rhythmically. So in normal rhythm, you can see the signal starts here in the right atrium, which is this chamber. This is the heart cut longitudinal, and then propagates to the two upper chambers of the heart, the atria, and then to the real pumps of the heart, the ventricles, where it's squeezed out for the rest of the body. And if you want to check that it is really an electrical signal that is coordinating this, you can put electrodes on the chest and you get a signal that looks like this, the ECG. You can have perturbations to this rhythm. The most common is something called atrial fibrillation, where the atria, the propagation in the atria is irregular, goes back and forth, and leads to this kinds of signals. So if we were to model what's happening all this using a biophysical model, we can go back to the Hodgkin-Huxley model. We need to make some changes because the shape of the action potential is slightly different. And given that we don't have just one cell, but an ensemble of cells, and the V needs to go from one to the other, the potential difference, as I've just shown you, we add this term, which is a diffusion term, if you're familiar with the diffusion equation, and D is the diffusion tensor for the propagation of the action potential. So this type of equation enables traveling wave solutions, great, which is exactly what we expect to see for healthy propagation of the action potential. So red being this part where the potential is high, and then going back all the way to resting state, which is blue. But it also enables you to model what happens in disease. So this is an experimental recording using a technique called optical mapping, which I'll go back to later, of fibrillation. And you can create fibrillation or a model thereof using computational models in the same way. See here, there's a source of fibrillation that moves around, which are often called rotors. So using this model, you can study all sorts of different processes. So this is a fibrillation in this strange shaped things are the atria only, and you can initiate some arrhythmia there. Oops, sorry. And you can initiate some arrhythmia there. And you can also, apologies, something wrong with the slides. And you can also, trust me, because the video is not showing, you can also make some changes to the properties of the system, mimicking the delivery of drugs such that these abnormal waves disappear. Other things you can do are create personalized models of the heart, so what we call digital twins sometimes, where here is the left atrium of different patients, 1, 2, 3, up to 6, different shape of the left atrium. These are models from MRI with different, and here this colorful patches are areas of fibrosis or scar, which are thought to play an important role in the fibrillation dynamics. So things you can do is, given a patient's geometry, the fibrosis distribution, and other information that you may know, you can create a personalized model, and then you can test different treatments. So here, it doesn't matter what these stand for, but PVI plus L, etc., for each of patients, and to try and understand which ones of them are most likely to succeed. And you can do this before, of course, you actually perform the treatment on patients as treatment guidance step. So these are the success stories, so to speak, of biophysical modeling and what they've been used so far. The alternative approach, data learning, just, I'm sure you don't need any examples for this, but just to give you a few from this type of cardiology problems. This is a set of dynamic MRI images called CINI MRI, and you can use convolutional neural networks to segment the cardiac structures here. the right atrium is not segmented, but this is the left atrium, and these are the left and right ventricles. And you can estimate the volume of the chambers across time. So here, this is the volume of the left atrium, estimated automatically. You can find the points of maximum and minimum volume to calculate something called the ejection fraction completely automatically, and use that as a biomarker for treatment guidance, which would otherwise be very time consuming to estimate. Likewise, you can be a bit more ambitious, and you can see if you can find out from, these are MRI images where it's believed that you can see the fibrosis in the heart that I was showing you before, and you can use this as inputs to your convolutional neural network, together with perhaps some other data about the patients, to try and classify them into needing treatment, not needing treatment, having a given pathology or not. So my question is, can we combine the two approaches? How can we make use of the advantages of both of them to solve clinical problems here? And there are a few examples of ways of doing so from the literature. So neural networks have been used for physics equation discovery. So you throw some data at the network, no idea what the equations are. This is an example from a few years ago. I think there's now a newer version. You can perform some analysis here, mimicking the thinking processes of a physicist to try to learn the actual expression that ties the data together. You can also use neural networks for model simplification, using Cindy and their derivatives and their other versions. This uses an autoencoder that simplifies the equations to a reduced state, and uses this reduced model to model what it is that you want for your dynamical system. And you can use physics-informed neural networks, which is what I'm going to be talking about for most of the rest of the talk. So you can use physics-informed neural networks, which are called PINNs from now onwards, for two main objectives. You can use for parameter estimation. Those are the parameters in your network, which you often, in your set of equations, which you often don't know and are very hard to estimate using other methods, or to solve the equations themselves. So if you remember this diagram from before, you can think of the PINNs as bridging the gap between the modeling, for which you had small amounts of data, by using some intermediate value of data, or perhaps even the same amount as in biophysical modeling, and having some knowledge of the physics of the system, but perhaps not as much as for the modeling itself, but definitely less than for the neural networks, and definitely less data, definitely more knowledge than for the neural networks, and definitely less data than you'd need for a pure approach of neural networks only. So the physics is here in the architecture used as a regularizer, and it's been used across lots of different areas in medicine, typically linked to blood flow, which are the examples that I'm showing you here. So I'll just use this Berger's equation as a motivational example to explain to you how PINNs work in a little bit more detail. So the inputs to your network are the coordinates of the system that you want to solve. So for here, it will be x and t, which are the coordinates of the points in which you want to compute a solution for this equation. And the solution will, of course, be called u. So you feed it to a neural network. Traditionally, fully connected neural networks have been used, like you see here, and I'll talk a little bit more about the architecture later on. And these will estimate your solution, your U. And then you can check whether your U matches your experimental data. That's what you would do in any neural network using a suitable loss function like the mean square error. For the PINNs, you also compute the derivatives of U with respect to your x and t, because that's what you do in your differential equation, and you can do that using automatic differentiation. It's just like the same backpropagation that you use to train the network. So using the chain rule. And so once you've computed those, you can create another loss function term. So here it will be a matter of just putting all this term on the left-hand side, and then you'll know that the residual should be 0 for that. So here, this sum of terms should be 0, and you can feed that as an additional term to your loss function. So your loss function will have two main terms, one for agreement with the experimental data that you have acquired about the system, and another one about agreeing with the known physics law of the system. And you can set these weights to 1 in the simplest example, or there have been a few other examples suggesting schemes to calculate the optimal weights for this. Typically, you solve differential equations using boundary conditions or initial conditions if there's a time dependency. So you also include them in your loss function as well. And I'll talk a bit more about this at the end. I've already said that the derivatives are calculated using, excuse me, automatic differentiation. And you can train this using synthetic data, for example, from the solver of your biophysical models, your digital twins, or you can use experimental data as well. This is increasingly done, but I would say 95% of papers do not use experimental data with PINNs yet. So why would you take the trouble of doing all this? So you can use the PINNs in two different modes. In the forward mode, you're using the PINNs as a way of solving the, finding the solution to the differential equation that best matches the data that you've acquired. And you can do, and it ends up with a solution with an arbitrary precision. What I mean by this is that although it's not an analytical solution, it works in the same way in that you're not restricted to having the solution only in specific data points with a spatial step or a temporal step separating them. And to do so, it optimizes the weights and biases of the network according to the loss function. So you can use it to replace your typical differential equation solvers, finite differences, finite elements, and you have the solution across the entire domain, as I've just said. Alternatively, you can use the PINNs in inverse mode. And in this case, you do not tell the PINNs the specific value of one parameter or more of your system. So here, for example, you could withdraw the knowledge about this parameter nu, and then the PINNs would treat the nu as yet another parameter that it's optimizing for when it does the training process. And it would optimize the weights and the biases of the networks and the withdrawn parameters for the equation at the same time. So it will provide you with a means of estimating unknown parameters such that you obey the equation and you're in best agreement with the data as you can. So this is an example from this paper where they use the Navier-Stokes equation to predict the pressure in this rectangle, and at the same time to predict this parameter here, which you can see was 1 in the original equation that from the ground truth, and these are the estimates from the PINNs, likewise for this parameter here. So the question I'm going to answer now is whether we can use this framework to make inferences about cardiac electrophysiology. I won't talk a lot or at all about the medical motivation for doing this, but it's ultimately to characterize what is happening in cardiac tissue from measurements that we have. So we have our measurements, typically clinicians look at them and make a diagnosis, say, oh, I see evidence that something is happening. This would be a quantitative way of doing so, and it would go beyond this assessment in the sense that it combines lots of different data in a way that's not amenable to human interpretation. So this is the paper, and we also have all the code that underpins what I'm going to be showing to you in GitHub. You can use that if you're interested. So the model we're using for this, the biophysical model, that is the equation that is the basis of the PINNs, is called the Aliev-Pamphilov model. It's the simplest model you can have to simulate cardiac electrophysiology. So this is the equation I've shown you before. So you have your potential across the cardiac cell membrane. This is your diffusion term to allow it to propagate to neighboring cells. And this is what I called ion before, so the model explaining how V changes by letting ions in and out of those ionic channels. So apart from V, there's another variable, W. W is a latent variable. You cannot measure it experimentally. And the reason it's there is to enforce the refractory properties that I was telling you about. So here in blue, we have V across time. This is the shape of the action potential from this model, which is very similar to the shape of the action potential in a ventricular cell. And here towards the end, you have W, and this is set up such that you cannot re-excite the cell and initiate a new action potential if W is at a high value. So as I said, this is the simplest model you can have, only two variables, V and a latent one, W, that takes into account this refractory period, that is the restitution properties of the model. It's differentiable, and it has these three parameters that our network is going to be asked to estimate later: the diffusion coefficient, scalar throughout, so comes out of this bit here. A, which is linked to the excitation properties of the cell, and B, which is linked to the duration of the action potential, so whether it's longer or shorter. And so these are the parameters that the network will estimate later. So first, I'm going to purely work with synthetic data. So this is created using a finite differences solver in MATLAB, in GitHub, in 1D. When I say 1D, I mean just a cable, 2D, a rectangle, and the three modes that we use for the rectangle. So you have a planar wave, which propagates here from left to right. You can have an excitation point here, and then a wave that is centrifugal. Or you can create one of these reentrant waves similar to the ones I've shown you before as modeling the arrhythmia. And this is the architecture. Again, it's very similar to the architecture I showed you before for the Burgers model, but now we have two coupled equations. So we use the same neural network to estimate both V and W. So we're assuming we can use the same weights and biases throughout, except here at this last layer. And we have two loss function terms. So one for the agreement of V and another one for the agreement of W with the equation. So putting it all together, it looks a bit menacing. Apologies. Agreement with the data, agreement with the equation for V, with the equation for W, with the boundary conditions, and with the, which here are no flux, and with the initial conditions, which are known. So we use the same architecture across all problems. There's an exception later on that I'll mention to you, but we've adapted the capacity of the network and the training scheme depending on whether we went to 1D, 2D, estimating one parameter or more. And throughout we use this library, DeepXDE, for in Python. We use the we use the TensorFlow backend. And it's very useful, I think, if you're thinking of solving these problems. I'd recommend you have a look at it. So the training time ranges from 15 minutes to 7 hours using one GPU, depending on the complexity of the problem. So just a couple of tests. As I told you before, W is not measurable experimentally. We can measure it, so to speak, in our in silico data, but it wouldn't be measured experimentally. But thankfully, the network can estimate V and the V time course perfectly without any data for W. So all the results I'm showing you next do not have any data for W. There's no data agreement for W. W is only enforced by the equation. The network performs well in the presence of noise. As you see, it copes with relatively large amounts of noise. This is 0.5. For the standard deviation of the noise, and you can see that the amplitude of the wave is 1. So it's quite a high value of noise. These black bits are the experimental data that the network is seeing, but it can still fit this very well. Note that this is just for one, this is across time, and it's just for one point in space. It will also have experimental data from other points in space that I'm not showing you here in this graph. And you can also really go down on the number of points that you give to the network. We went down to about 100 points before it performed poorly for this 1D cable simulation, which was in a cable of 10 centimeters running for 50 milliseconds, sorry, not seconds. So this is the planar wave simulation. Here we're trying to see how well the pins can solve the equations, have the ground truth with the finite differences model on the left, and the pin solution on the right. And I always have to double check that the right videos, because they look exactly the same to me. Likewise, this is the centrifugal wave. So starting from a corner, it propagates. There are some tiny differences, but they're very hard to identify. And this is for the spiral wave, the rotor that simulates an arrhythmia. And here you see more differences, particularly near what we call the tip of the rotor, this area, which is where the wave curvature is highest. And things change more rapidly spatially, if you want to call it that, but it still completely captures the dynamics. If you want to use the pins in inverse mode and find the parameters of the system, it also works quite well. You can see here estimates of B, which was related to how long the action potential lasted, are particularly good. And throughout, it performs better for the planar and centrifugal waves than for the spiral wave, as we saw in forward mode as well. Estimates of D are also quite good. Not so for spiral waves, and I can, if you're interested, I can elaborate on why I think that is later on. And for A, it also does a decent job, particularly again for planar and centrifugal waves. You can estimate them in tandem. So here you're estimating D, but you also estimated B at the same time. And then it performs slightly worse when you're just estimating D on its own. But the parameters B, A, and D are quite decoupled in the system. So although it performs slightly worse, it's not bad. And we throughout, even though we are estimating the parameters that the system doesn't know, it's still able, as you can see from these figures, to reproduce the morphology of the action potential across space and across time. In the paper, you'll see that we also did these experiments in the presence of variable amounts of noise, and that the estimates hold up well, particularly for this B parameter related to the action potential duration. So on that note, we wanted to see how robust this setup was when used with experimental data, where our model wouldn't really capture all the details of it. So we did a cross-model experiment. We kept the simple Aliev-Pamphilov model as the model for the pins, but we changed the generating model of the ground truth. And we made changes to the generating model such that it generated action potentials with different durations. And then we fitted it with the pins to see if it could pick up these changes by changing the value of B. So a shorter action potential, as you see here, 158, 98, 74, corresponds to an increase in B, which the network duly captured. And it was able to fit the action potential morphology quite well, except this bit here, we made it smoother than the actual model contemplated, although again, the data it saw was very sparse and is represented by these circles here. So then we also tried to see if the pins were able to identify heterogeneities in the model. So keeping the same rectangle, we introduced this lesion here, which is this square here, where the diffusion coefficients was a fifth of the value for the outside, for the baseline value. And because the diffusion coefficient effectively controls the propagation speed, here in this lesion area, you see the propagation slower, which basically leads to this bending, so to speak, of the wavefront, in the centrifugal wave, in the planar wave, and in the spiral wave. And so we modified the architecture For D, we did not assume that we could use the same architecture and just cope with the change of the weights at the last layer like we did for V and W. We used a different one, same inputs. D doesn't depend on time, so time is not an input here. And we use this sigmoid here as a bit of a trick, because we're only expecting two values as an assumption, the healthy and the unhealthy D. So we weren't asking the network to solve the much more difficult problem of what is the actual value of D. We were in fact asking it, is it healthy, is it unhealthy for each point in space? Everything else remains unchanged, and the D will of course contribute to the, as a part of the equation, to the loss function here for the equation of V. This takes much longer to solve because the architecture is much more complex, much more to fit, so up to 16 hours using the same setup as before. So it works well, as you can see here. These are the estimates of the network for the centrifugal wave. You can see that it identified the region with low D quite well, some slight inaccuracies towards the edge. Likewise for the spiral wave, though it struggles a little bit here close to the tip of the spiral wave as well. That might be because we only had 50 milliseconds, apologies, of simulation time for training data. I think if we had a longer time course, then the tip would move across space and this would probably disappear as well. And you can also do this and estimate the other global parameters, the B and the A, at the same time, and those are well estimated as well. So then we decided to train with real experimental data. We used optical mapping data, which I've shown you before. So these are cells from some ventricle of rats, and you prepare them with a dye with a dye that depends on the transmembrane voltage. So here's your setup. These are very high spatial and temporal resolution images. And we had pairs of images baseline with no drug and with a drug called nifedipine that shortens the action potential duration, and then another set of control data and another drug, which in this case increased the action potential duration. And we wanted to see if the PINNs were able to detect these changes in APD. So here's a snapshot of one of the action potentials used. We used much more experimental data here than we had for the in silico tests. This is nifedipine that decreases APD, and the PINNs are able to detect that and see a significant difference in B. For this drug, the PINNs don't see a significant difference, but only looking at it, there's not much of a difference in action potential duration. So I think that's actually a good thing. They're not really finding differences that are not present in the data. So this opens up the idea that the PINNs can be used to identify the effect of drugs on these action potentials with quite a lot of detail. And that's something we're exploring. So I'll tell you a little bit about the future work that we're doing here. So I've just shown you that we can use this PINN setup to simulate cardiac electrophysiology and to perform parameter estimation. I repeat that we did not use any data for latent variables, which wouldn't be available experimental at all. And we used that simple architecture that had the same network for V and the latent variable W. In the meantime, we've also found that we can use the same setup without the Aliev-Pamphilov model and with a model that's much more detailed and that has more than just one latent variable. And those different variables and different terms can be associated with different ionic channels. And so that, again, recapping what I've just said, allows you to identify which of these channels the drugs may be working with. And it can also actually we found that it doesn't need to know the equation even for W. It performs well if W is a freely varying variable too. And I can talk more about this later if you're interested. So I've also shown you how we can use it to work out what the effect of antiarrhythmic drugs is. And I've just said this also works well with more complex models. we use much lower amounts of training data than if you were to use a neural network without the PINN's loss function on it. But a detail of the PINNs is that it needs to be trained for every set of experimental data. You can't just, the network itself doesn't know how to uncouple the equation from the data. It's all bundled up in the loss function. So if you were to acquire a new set of fresh data, although you have you've trained the PINNs on another data set, you have to train it all from scratch, which is a quite time-consuming process. We found that we can shorten that training time if we pre-train the models with other data, and we're still trying to work out what the best way of optimizing this is. So we found that the parameter estimation was good for B. For A was worse in the presence of noise, and also worse for spiral waves. And I think this comes from the fact that the data isn't really ideal for the estimation of A. Again, I can elaborate on that later. And it comes from the problem that I've just mentioned, that you can only train PINNs on one set of experimental data. So even if you have different experiments for the same model, you cannot combine those easily into the one loss function that the PINNs needs. So we found that it can cope with experimental noise well, and we've also found in very recent data that it works well in 3D. Our heart is shaped like a spherical surface in 3D, but we'll slowly move it towards a more realistic heart. Use it to identify spatial heterogeneities in a model parameter, and this is probably the easiest path towards clinical application. That reduction, apologies, in D is what happens in this fibrosis of scarring that I've mentioned before. And if we were able to get this to work with clinical data, it'll be a really interesting tool to identify whether there is fibrosis and roughly whereabouts it would be. And for that, we'd of course need to make inferences from signals from electrodes rather than this optical mapping for data for V, which is not available clinic. And this is an area we're working on very intensely. And if you ever invite me again, I'm hoping to be able to tell you how we've worked out how to make inferences from this data, and also from ECG, so from data acquired from the chest rather than directly on the surface of the heart. And latest, another question we've been working on is whether we can use a different network architecture for the PINNs. As you remember, we've been using fully connected neural networks. Recently, there's been an architecture called DeepONet that has been proposed. I'll show you a little bit about it. This is the paper, and this is the architecture. And it was originally created as a means of approximating mathematical operators. So you'd give it a set of data for you as your inputs, and then your output, which would be the result of applying this operator on your X in here called Y. And then you just train this network such that you'd be minimizing the mean square error between Y and G of X, if that makes sense. And so if you do that, and they propose slightly, this architecture with a trunk net for Y and the branch net for, sorry, I said X was the input, but it's actually U of X, U being the function of X and X being the positions. And they propose a different architecture with this trunk branch network for this particular application. As I said, you still need the numerical solver to generate the training data, this Y here, but the idea is that you learn a neural network-based way of approximating these operators, and then they have examples like Laplacians, other stochastics operators, antiderivatives, and so forth. And as I said, use a simple mean square error loss. And then after that, another paper has been proposed that this DeepONet can also be used for PINNs and performs better than the PINNs, than the fully connected network we've been using. Actually, it performs so much better, the authors claim, that you don't even need to train it on any experimental data. You can use your boundary conditions and initial condition losses as a replacement of the experimental data. So this would be a way of training the PINNs without any experimental data. And they show convincingly that it performs better than the original PINN. So this, again, is the same loss function as in the normal PINNs, but the network architecture has been replaced by this DeepONet. And if you're able to do that for a range of problems, then you would, because you don't need any data, you don't need your forward solver at all. So this would be a way of creating basically another solver altogether for differential equations, bypassing finite differences and finite elements. It's not clear whether this would also work in inverse mode, and that's something that I think would be really interesting to look into. So I leave with that point. I'm not sure if I have have time.
Yeah, I think you're okay.
I do? Okay,
yep.
Great. So I'll just carry on with that. So this is a slight different change of tack. This is telling you a little bit about a simulator for generating cardiac MRI.
Sorry, Marta, you're not sharing the screen.
I'm not? Oh, apologies. Yep. Thank you for letting me know. So I'll share the screen again. So this is a simulator for MRI data. So it's a synthetic data simulator that I'm hoping to use to create data to use for the PINNs afterwards. But I'm showing it to you in case any of you has an interest in it. And if so, please do get in touch and we can look at how to use it best. So it's actually based on a MATLAB GUI that was proposed a long time ago, and I don't think it's maintained anymore. And this is the link to the MATLAB GUI. What we did was basically port it into Python and allow batch creation of images. So this was a GUI, so you set up the parameters, generate one single MRI image, and now in our version, you can generate batches of images with small variations, and the idea being that it can be used to, for example, train neural networks. So as inputs, we have a phantom with MRI tissue properties. These are the very most basic ones, but there's scope for incorporating others. And we put it in our MRI simulator that knows parameters about the sequence you're using. Again, basic ones in here, TETR, FA stands for flip angle, and other ideas. And then you can generate several scans with different variations. And the application I'll show you now, this data was used as inputs to a neural network. And the aim was to see if we could train the neural network on these simulated data and then test it on, ambitiously, test it on real patient data. So bypassing the need of manual annotation of the patient data. So what we did, the task, I'll show you some data in a little while, was a direct estimation of the volume of the left ventricle, of the muscle of the left ventricle, using this 3D ResNet without explicitly segmenting the left ventricle and then counting how many voxels were labeled as such. So this is the phantom. So it comes from this phantom proposed by Zegers et al. Called XCAT. And this is nerves based, so it's based on splines, and it's got embedded a way of moving the splines around so that you can change the shapes of the organs without them all overlapping in realistic modes of variation. So we took that and we assigned realistic MR properties to each of the tissues. Here I'm showing you the T1, which is the longitudinal relaxation time constant, if you're interested. And this is a transverse relaxation time constant of the tissues. And we added some intra-tissue variability by letting the values vary across as a Gaussian distribution for the same tissue type. And so these are the images. No prizes if you guess which ones are the simulated images and which ones are the acquired images. There's slight difference in contrast. We're able to vary the anatomy quite a bit, namely the cardiac size, but one thing we can't vary is the orientation of the heart, which is actually quite variable from person to person. So what you see in the surrounding the heart, so here's your heart, This is the a 4-chamber view that I've shown you before when I was talking about the arrhythmias. So you can't change what organs you see next to them, which is something that we're trying to look into. And these are different slices across different levels of the heart. So still no heart here, and this is the close to the tip of the heart, the apex, and then moving here towards the atria and the top of the heart. So, so far, we don't actually have much flexibility on the MR sequence design. That's something we're working on, which is why the contrast is slightly different. And there's a huge list of things that the simulator doesn't include yet that we're trying to put in, and that I won't go into the details of. And I've already told you that we struggle with the heart orientation, and also it's difficult to introduce pathological changes as well. For example, if you were to add a tumor somewhere, it would be quite tricky to do that realistically. So for our proposed task, if you train on simulated data and then test on the simulated data, as you can see, this is the true ventricular volume, and this is the predicted one here. It works really, really well. If we start if we train on real data and then test on real data, it doesn't work so well. It's quite a challenging task, and I think our network might need to have its capacity improved as well. This is really preliminary data. And then here, if we use the simulated data to help the training, it actually does help the training, and it gets closer to what the identity line, which is this red bit, and here's in blue is our best fit line, but it still doesn't do an amazing job. So as I think the neural network is underfitting slightly, and we're looking into that, and we're also, I think, we're simplifying the task a little bit, and we're now trying a task where the network just says, is there a heart or is there a left ventricle in this slice, yes or no? Having said all that, if you look here at the bland Altman plots for this task, it's not as bad as these graphs seem to suggest. There's literally no bias in the volume estimation, it's the variability that is quite high still, but this is work in progress. So again, hopefully it will work well. So the aim of this is not to train these relatively simple networks, although that wouldn't be a bad thing, is to use it as a platform in which we can test MR reconstruction neural networks. Neural networks from MR reconstruction are typically supervised, and this would be, again, a way of bypassing that need and making them unsupervised. And I'm dying to try to use them in a PINN setting and see if we can estimate the biophysical parameters or some of the biophysical parameters of the tissue directly, which would be a very, very complicated equation, but using the PINNs setup. So just to conclude, I hope I've convinced you that PINNs bring the best of both worlds and get some of the benefits of biophysical modeling and from data learning together, and that we can do that for the understanding of cardiac physiology and pathophysiology. And as we continue this line of work, hopefully we should start seeing a clinical impact from this. I think the lowest hanging fruit is the estimation of this fibrosis in the heart from electrical measurements that I've been telling you about. Just very quickly, wanted to thank the students and the academics that contributed to this work. And as I said before, I'm very happy to answer any questions you may have.
Thank you, Marta. I really enjoyed your talk. It's very relevant to what I'm doing as well, even though it's like different field, but the applications seem quite similar. So you have any questions for Marta, you can type them on the chat. I can read them out loud, or you can just raise your hand and yeah, I'll give you. There's a question on the chat. It says the XCAT is not a free data set, right? And is it possible to simulate some pathologies or different scanning sequences?
No, the XCAT is not is not freely available as far as I know. So ultimately, if we do want to make this freely available later on, we'll have to reach an agreement about that. It is possible to simulate some pathologies, for example, hypertrophy or dilation of the cardiac chamber by changing so hypertrophy would be an enlargement of the cardiac muscle, and we can do that, and the dilation pathology would be a dilation of the blood pool. Yes, we can do those quite well, but we could have done that in the regression task I was showing could have been done in a classification way of healthy pathology, but we wanted to keep it as a regression to make use of the full data, so we didn't do it like that. Different scanning sequences are definitely possible. We've been struggling a little bit with that. So we've been basically using the ones I've just shown you, but there's scope for expanding it for sure.
Yeah, thank you, Marta. Anyone else have another question for Marta? In the meantime, when you were speaking about the MATLAB approach to generate more images, you mentioned that it was constrained to only one orientation of the heart. Is there a way that you can generate more data using the data that you have and account for that?
I was a bit unclear with that. So what I meant was not that we were constrained with the orientation of the imaging plane. So I showed you 4 chamber data in that particular orientation, but we can rotate it, translate it to whatever we want. What is tricky is the angulation of the heart within the body. So the heart is the apex of the heart is pointing kind of left and down, but in some people, but the angle relative, say, to your axis varies a lot from person to person. And what that means when you're doing the MRI images is that the organs surrounding your heart may change. In some person, you'll see a lot of stomach. In others, you won't see, it's just lungs. And that's the bit we've been struggling with. But the actual imaging plane, we can choose whatever we want, as you would in an MRI scan or a real one.
Will you be able to use some kind of like generative network to create more data and to account for the change of the angulation?
Yes, we could, but we've been trying to avoid it because we're purists, basically, and we wanted to have it all physics-based so that we could then change a parameter and know exactly, basically do derivatives of how changing this parameter affects our network performance and so forth. So we've been avoiding generative models, but yes, we could. I think moving forward, we'll probably have to combine them to get more variation in the data than we're capable of modeling with purely biophysical approaches now.
Yeah, do you want to go?
So yeah.
Can you hear Rosella?
Can you hear me?
Yes.
Yeah, I was just commenting, saying, what about a physics-based generative model?
Yeah, I haven't explored those at all. I'm not even sure I know much about them. So you have a GAN and you have the physics constraining the GAN. Basically, yeah, I think that would be really interesting to explore, but we haven't really done so yet. We're sticking to, as I told you, one of the aims would be to then put this data into the PINNs and see if we can do that to find MRI parameters. So we're trying to be purists in that respect, but yeah, I haven't looked at that yet.
Thank you, Marta. Yeah.
Actually, I have a question, let's say, probably completely unrelated from the technology and the models behind that. But I was just wondering, how do you study the impact of a drug?
So it's what do you mean, impact in what way? In a specific person or
yeah, so what you are simulating, you said at some point at the beginning of the presentation, we use that for understanding the impact of a drug, if it's working. So I was obviously, in my mind, as I'm not expert of that field, I was thinking probably the blood pressure, stuff like that. And I was like, what are they simulating actually?
Yes, so that's a really interesting question. The drugs I was specifically referring to are called antiarrhythmic drugs, and they're drugs taken to terminate an arrhythmia or improve an arrhythmia. So the success is on a given patient is whether they led to a termination of an arrhythmia or an improvement of symptoms or not. There's a huge danger in that actually, if you give the wrong drug, you may make the arrhythmia worse. Which is one of the motivations for personalizing the drug use. You should know exactly whether that patient is suitable for that drug. The application I'm thinking about when I'm doing these things is slightly different. So the drugs are tested in the lab during development on single cells, usually, and then you see the changes in action potential properties that I've shown you. But there's not really any testing to then see what the effect is at the whole organ level. So that's part of the motivation, both with the modeling, where you can model those changes in the whole organ, and also with the PINNs, where you can then do the measurements of the whole organ and see whether the effect is or not. And you know, the first order, you could think, oh, the effect's the same, the organ is just a group of cells, but actually the way the cells interact with one another affects the way the drug works. So that's why that's an interesting problem. I hope I answered your question.
So, of course, yeah, you answered my question. And so I was thinking, this means that combining your model, like your approach, the PINNs with data learning, potentially you can understand the impact of a drug on a specific person before giving the drug to that person.
Yes, if you have the right data. Yeah.
Amazing. That's great. Thank you.
Thank you, Marta. If we don't have any more questions, I think we have time for one more. Yeah, otherwise we can leave it up to there and like say thank you to Marta for an amazing talk. Great start of the year as well, and very, very inspirational.
Thank you very much. Thank you. Very nice speaking to you. Bye-bye.
Yeah. Thank you. Thank you, everyone.